Welcome to the lecture series on vitamins and nutrition. In today's session, we are going to continue with a water soluble vitamin, folic acid. Today's content would be on the sources of folic acid and its mechanism in which how folic acid gets into our system. So, we are going to look at the absorption parameters of folic acid, and we are also going to look at how different sources give rise to differences in absorption and the dietary needs of folic acid. This part we have covered little bit in the previous session when we spoke about the historic content of folic acid and its metabolic role. We even came to the level of discussing its metabolic role, but it will be more appropriate to start from the absorption aspect of it in detail, so that gives you connection between the deficiency conditions which are associated with folic acid, because some of the deficiency generally would arise from defects in absorption or processing in absorption and problems in the distribution of folic acid. Then we are going to go through further on the metabolic role of folic acid, how it contributes in various pathways, especially in the synthesis of various components, how folic acid directly plays a role, and we are going to finish off with deficiency conditions of folic acid. So, let us go back to the sources of folic acid. As I have told you, folic acid being a water soluble vitamin can be taken from variety of sources, especially the green leafy vegetables, as you look over here. So, green leafy vegetables is a very good source of folic acid, and the total body store would be somewhere around 0.5 to 20 mg. So, let us say you stop taking folic acid for a long period of time, your reserves can last up to 6 months. Okay, so it can stores the stores of liver and other different tissues in your body that would contribute in the folic acid reserve could actually protect you for a considerable period of time. So, what they have done, giving way for it, the government have made sure that folic acid can be fortified in cereals and grains, because during processing and during various other operations, the food is likely to lose its natural folic acid storage, especially when you are going to cook the food, you might lose folic acid. So, considering all this, to supplement for the lost folic acid in the processing, government has recommended fortifying foods, especially processed foods, with folic acid, because folic acid is supposed to be a very important water soluble vitamin. So, this picture we have seen earlier. So, this gives you various sources. So, you see papaya, okra, all green vegetables, seeds and nuts. You find fruits, oranges and avocado, and primarily you look at all the green leafy vegetables. You have the tinge of color over there that contributes in folic acid. So, you might wonder, most of the B soluble vitamins, when we consider, they are part of the vegetable source, and as well as you do have other products available. Now, if you look at it, the recommended daily dosage. So, this we have seen earlier. I want to just kickstart from where we left. So, your daily requirement of folic acid keeps increasing as we grow, because new tissues, cells which grow and which have been replaced, which have been maintained, need folic acid. Okay, and you see here, very interesting enough, although we have seen this, we know that folic acid deficiency can have a major problem in pregnancy. So, during pregnancy, it has become mandatory that the pregnant women should be supplied with sufficient amounts of folic acid, and this can continue further in the process even during lactation phase, not only during gestation, but also during lactation folic acid would be required. Now, you look here how folic acid basically is utilized in different processes. So, you see folic acid is normally utilized in the folate cycle. So, you are going to see that shortly, and folic acid is actually a precursor compound, the real active compound being tetrahydrofolate or tetrahydrofolic acid. So, tetrahydrofolic acid is basically, can you see over there the tetrahydrofolic acid? If you consider the previous structure of folic acid, you would really understand exactly what this tetrahydrofolic acid is all about, because if you look into the structure of folic acid, you really appreciate exactly how the folic acid components get activated before they could be made available or utilized by our system. And also we are going to look at how folic acid is playing an essential role in the purine and pyrimidine synthesis, which are the primary raw material for a DNA. So you see DNA synthesis, amino acid synthesis, nucleoprotein synthesis, neurotransmitter like catecholamines, all are dependent on folic acid, and also increases the metabolism of formic acid. So you do have variety of requirements of folic acid in various stages of biochemical mechanism in human physiology. So we are going to look into the absorption profile of folic acid. The folic acid what we take through oral route, the dietary sources. So you see the plasma concentration normally in species reaches somewhere around 10 to 30 millimolar, which is in the free solution. So the predominant form in portal plasma is the reduced form, tetrahydrofolate. So that's the actual form you find over there in the plasma, and this is being taken up by the liver, where it is released to the peripheral plasma after converting into 5-methyl tetrahydrofolate, or also to 10-formoyl tetramethylfolate. So these are some of the activation products of folic acid that is available in our system to be made use during the normal metabolic process. You see here the absorption of folic acid is quite dramatic. You see there tetrahydrofolate reductase, tetrahydrofolic acid over there, this particular enzyme doing this conversion process. So you see there the total folate is like 5 to 10 mg in human body, and most of them, let's say half of it, is present in the liver. Okay, so in the liver it is present as tetra, penta, hexa and heptaglutamate. So the last ring in the folic acid, so this is the ring, if you could recollect, the glutamic acid ring. So you do have polyglutamic acid residues. So you can have, that's what is mentioned here as tetra, penta, hexa, hepta up to 7 of 5-methyl tetrafolate and 10-formoyl tetrafolate. So this is how liver stores or utilizes folic acid. And you also see folic acid going inside erythrocytes. So greater concentrations, let's say 50 to 100 nanomolar of folic acid is also present in RBCs. Okay, so these stores are accumulated during erythropoiesis. So means when RBC is maturing, they all get inside the RBC and are made available in the process of RBC. So once an RBC is mature, its uptake is limited. The uptake of folic acid inside mature erythrocytes or RBCs are limited. If you look at some of the factors that might have an impact on folic acid, we will see some of these points also discussed during deficiency. So you see there cigarette smoking and chronic alcoholism. Okay, so people who are into these habits have an impact on the amount of folic acid that they consume. Already the liver is at danger because alcohol and other components, the xenobiotic components which reach liver, can have a detrimental effect on the liver metabolism. In addition to that, you see folic acid transport is also reduced, so that could keep people at grave danger of developing folic acid deficiency quite rapidly, especially for chronic habituated people who have this smoking and alcoholism. So you see there for non-smokers, okay, so the plasma folate levels can be nearly half those of non-smokers. So you see people who are having a normal healthy diet and people who get into habits like this, the kind of impact that can have on their metabolism. So most of the folic acid bound protein, okay, is likely to be folic acid. So it means there is this albumin and folic acid binding protein that transports folic acid in the plasma. So you see that the bound version is supposed to be folic acid in nature, and then you have unmetabolized folic acid. So, you see this level of unmetabolized folic acid, especially people who are consuming large doses of folic acid prior to folic acid deficiency, okay, or consecutively small oral doses who are taking folic acid way back in sufficient amount, could have been part of deficiency therapy or maybe as part of the dietary plan that they are taking or consuming more amount of folic acid. In any case, if it amounts 100 microgram or more, you always detect unmetabolized folic acid, or the folic acid what you detect to govern the plasma circulation would always be within the acceptable limit. But for people who consume larger amount, you find lot of unmetabolized folic acid in circulation. So, this is the picture. So, the absorption happens in the proximal small intestine, the duodenum. Okay, so, dietary folic acid, if you consider almost 50%. Okay. So, the bioavailability of dietary folic acid, although present in the food substances that we take, but eventually processing can or oxidation can reduce the availability. But when taken directly as folic acid from supplements, the bioavailability is quite 100%, because you do not have much barrier problems connected with folic acid, where folic acid could be made available in the tissues. And as we saw just now, how folic acid is metabolized in the liver, and unmetabolized and metabolized folic acid are excreted in the urine. So, if you quickly look at the summary of what we have already done till now, so you would know that it is a water soluble vitamin. We have designated this to be B9, and all the green vegetables, okay, leafy green vegetables, that is why the name folium, which actually become folic acid later. So, you see there the duodenum is the place where absorption happens, and you see they are essential for synthesizing purine, pyrimidine, methionine, catecholamine and all these components. And also if you look at the daily requirement actually increases, and in the pregnancy and lactation you see there is even more high demand for folic acid, primarily for the metabolic function happening in the mother, as well as folic acid being utilized in great amounts by the developing fetus, because developing fetus would need more amount of folic acid as part of cellular proliferation which is happening inside the fetal tissues, would also require more amount of folic acid, and the immediate source of folic acid would be from the mother. So, mother has to take lots of high volume of folic acid to really meet the demand of the growing fetus as well, in addition to her remodeling of tissues in association with gestation. So, you see there what we have saw in the previous session, as we are kickstarting from there the overall bioactivation of folic acid. So, you see there this particular enzyme dihydrofolate reductase, which actually converts folic acid into dihydrofolate. So, there is one more conversion happening which gives rise to tetrahydrofolate, and then you also have fundamentally addition of glutamate, tetrahydrofolate synthase, so that tetrahydrofolate, the glutamated version, would be now available, bioactivated version for its function. So, you see there what would be the use of folic acid, exactly what folic acid is contributing in the metabolism. So, the active form of folic acid, as we call it as tetrahydrofolic acid, is produced from folic acid by an enzyme dihydrofolate reductase. It is a two-step reaction requiring 2 NADPH. So, you see the carbon unit carried by tetrahydrofolate is bound to nitrogen or N10, N5 or N10, or to both N5 and N10. So, that is a transition that starts happening. So, tetrahydrofolate allows 1 carbon compounds to be recognized and manipulated by biosynthetic enzymes. So, this is where the action starts, where 1 carbon compounds to be recognized and how they can be manipulated by a biosynthetic enzyme to really make folic acid participate in various metabolic actions. So this is a single carbon unit carried by folate. So you see there are a lot of things, the 5-methyl, the 5-formoyl, 10-formoyl, 5-10-dimethylene, 5-10-methinyl, 5-formimino, all these are like single carbon reactions. So you see there, this is the active zone where the single carbon units are carried by folate. So you have a very good collection of how this can proceed further in the deficiency diseases. Leave alone deficiency diseases, you are looking at all those metabolic processes where folic acid actively contributes, and obviously during deficiency all this essential function would be at stake. So that's why we really consider treating the patient as quickly as possible as soon as we have the first onset of folic acid. Otherwise it can really aggravate cellular tissue damage happening due to slowing of metabolism. So you see a bigger picture of dihydrofolate reductase. So it's all sequentially connected, and you see over there the folate N5 and N10, the single carbon reactions as we saw just now. Now this picture gives you a summary of exactly how tetrahydrofolate reductase could participate and contribute in various reactions. Okay, so you see that the dietary source of folic acid, the folic acid coming inside, dihydrofolate reductase converting folic acid into dihydrofolic acid, then you have dihydrofolate reductase again reducing it to form a tetrahydrofolic acid. So the overall bioactivation, if you have to consider how folic acid is actually converted into tetrahydrofolic acid, you see over there the folic acid is basically acted upon by dihydrofolate reductase in the presence of NADPH. So you see over there the double bond being saturated and you get NH, the dihydrofolate. So here you have the saturated version, and then you have a single bond being replaced, so you have dihydrofolate. Now again there is NADPH contributing, becomes NADP, where dihydrofolate reductase is acting upon. Now one part has been already reduced, and as you go further there is again both the parts are reduced. This is what we mean by tetrahydrofolate reductase, which is the enzyme which is catalyzing this reaction, and here you have tetrahydrofolate reductase, and as you see there tetrahydrofolate synthase would ensure a complete transition. Not only here the structures which are reduced to form dihydrofolate reductase and tetrahydrofolate reductase, you also see here tetrahydrofolate reductase is basically stabilized now. So if you could compare the structures, you would be able to really appreciate what's going on over there. And here tetrahydrofolates, can you see the n number of units, the glutamic acid units that can be added one by one. So you could be able to, you could visualize how this would have a tremendous impact as to how the activation process proceeds. So all this activation process requires enzymes, and each of these enzymes are essential targets in the normal function. So we are going to look at the 1 carbon, the carrier of 1 carbon units. Okay, so the carbon unit carried by tetrahydrofolate reductase is bound to nitrogen N5 or N10 or both N5 and N10. So this is the picture that we are seeing over here. So you could consider the 5-methyl, 5-formoyl, 10-formoyl, 5-10-methylene, and then you have 5-10-methylene, methinyl, and 5-formimidol. So methinyl and 5-formimidol. Okay, so you see that it all centers around this particular region, the nitrogen region from either side, where that contributes to the flexibility of this particular compound. So you see there how a single carbon units carried by folate would be able to get into the picture of a fine activation, a fine metabolic role happening. Now again we are going to zoom it up and see how this works. So you see there dihydrofolate reductase, and finally 10 formoyl derivative, and then you have a 5 formimino derivative, and here again you have 5 formoyl derivatives. So you see there, so in most of them it looks like it's a kind of a reversible reaction. So this is the main pathway, and as you see over there, thymidylite synthase, so converts dump uridyl to the DTMP. Okay, so you have this conversion is also linked over here, where we have methionine synthase. So from B12 to methyl B12, and this methyl B12 in turn converts homocysteine to methionine. So it's all a kind of a cyclic process. So wherever you work, your enzyme function as we are describing it can be linked with other enzyme functions. So it works in a cascade manner initially to study, but in reality we find many of these reactions occurring simultaneously inside the cells. So you see there we try to ensure the polyglutamate addition to the main compound. So here we have the folate and all the derivatives what you need to remember and practice, and how each of these derivatives contribute in the metabolism and function of the corresponding reaction that we are looking at. So we are going further to one more component, and this picture gives you the real source of what functions that we need to look for. This is again a schema of what we have already done earlier, especially in the summary part, and we are just expanding the idea further to look at how or in which step inhibition can be achieved, and this inhibition can have a tremendous impact on cancer cells which would need folic acid, because there is high turnaround of DNA synthesis, and instantly purine and pyrimidine synthesis will also be needed, and this is the raw material for building the DNA. So it's all linked as you see there. So the dietary source folic acid, it's getting converted to dihydrofolic acid, then tetrahydrofolic acid, which gets finally activated to N5 N10 methylene tetrahydrofolate. So this is the key compound, and this compound here you find catalyzes or participates in the thymidylate synthase reaction. So you see there DUMP is getting converted to DTMP. So this reaction is essential to carry out the function, and here again we have N10 formoyl tetrahydrofolic acid, which eventually contributes in purine synthesis. On the other end, if you look at the 5-methyl tetrahydrofolate, as it goes inside this cycle, you find homocysteine getting converted to methionine, and methionine gets converted into SAM, adenosyl methionine. So it's a kind of a cycle, and this one plays a very important role in the catecholamine synthesis. So you see there catecholamine synthesis, the overall function of tetrahydrofolic acid, how they are going to really contribute in the smooth movement of one metabolite to the other, how it can be thoroughly make sure whether other components in the metabolic pathway, especially the catecholamines, are synthesized. So you see there it's so linked, so very nicely linked, activation into the single carbon reactions being catalyzed, and that ends up in purine synthesis, and as you see over there you have catecholamine synthesis which is happening on the other end. Now we are going to look at little more closely the pathway what I have already told you, but in a different context. So many of the reactions what we are going to look at, you find the red ones tell you about the compounds which can inhibit these key enzymes. So the red ones are the inhibitors which can have an impact on the corresponding enzyme, and this in turn will slow down the utilization of folic acid by the cells. So, especially fast growing cells, cancer cells are supposed to be fast growing cells. So, there is an higher turnover of purine and pyrimidine, and this would be incorporated. So, the drug what you are using, especially the anti-cancer drugs, come under one special category, what we call it as anti-metabolites. It may not be directly connected, but we should be definitely be aware, because it is one of the essential vitamin in our body. So, you see over there, as you look at purine and pyrimidine synthesis, how they contribute in different levels, and all the red ones which are supposed to be inhibitors have their own mechanism in affecting specific enzymes in the pathway. And as you see over there, it might appear straightforward, but actually if you closely watch, so the entire network of pathways, as you look at, takes in attention some of the compounds which could compete or which could inhibit any of the processes shown over here. So, for that matter, you have hydroxyurea, gemcitabine, then p-methoxide, 5-fluorouracil, and all these compounds are playing a very excellent role in the activation process. And you see over there the cycle goes, and here the methionine being produced from homocysteine, and as homocysteine goes down as a cysteine. So, you see over there the cysteine, homocysteine, methionine contributing in the tetrahydrofolate pathway. So, that is one of the reason we need to look at how these drugs are closely related with each other, and little balance can completely upset the cycle. And it is also quite interesting to note how these discoveries were made in a very serial manner, so that systematically people could ascertain the use of folic acid in living cells. We are going to look at some of the enzymes, because these enzymes are involved in the acquisition of single carbon units by folates. So, primarily folic acid acts like a cofactor with a well known enzyme, and as a team or as a complex, it is able to cater to various enzymes involved in the reactions. So, let me read it to you, the lists, and it might surprise you the wide implication of folic acid in various metabolic processes. So, you see there the single carbon unit, basically the methyl group. So, the folate derivative would be 5-methyl tetrahydrofolate. So, you see the kind of enzymes that they contribute to. 5,10-methylene, you have folate, tetrahydrofolate reductase, and the methionine synthase. So, these enzymes are also being benefited from the folate derivative for its single carbon unit transition happening over there. So, you see there the list goes. So, you have a methylene group, you have a methinyl group, you have a formimino group, and then you have the formoyl group, and the corresponding compounds, the folate derivative, basically you could look at how we can modify the role of these individual derivatives and what are these enzymes that make use of this individual compounds. So, we are going to look at some of the inhibitors. So, aminopterin is one such inhibitor, and here you have some of the compounds that would be very important. But before we go on with this pathway, let us look closely about some of the components which are potential anti-cancer drugs. In fact, the anti-cancer drugs which act upon this particular pathway are having a very good impact in terms of the anti-cancer activity that they contribute, because it is a metabolite, and any inhibition of the metabolite could have an impact in the way how folic acid would be utilized. So, this is a very popular compound which actually is a very good anti-cancer drug in the treatment of leukemia. So, we are going to summarize the first session. We have seen the sources, we have seen the absorption pathway, we have seen the metabolic role of folic acid, and now as a summary I would like you to take down 2 questions. Describe the source of folic acid, describe the metabolic pathway in which folic acid would be activated so that your body and cells would be able to make use of folic acid for its metabolic function. So, if you could write down the pathway of how tetrahydrofolate is formed, so that would be the answer for this question. And further we are going to ask a question, how folic acid can be inhibited and how folic acid inhibition can be therapeutically utilized for developing this category of antimetabolite drugs. Thank you once again for your active participation.